All right, so in this lecture, we are going to start getting into kind of the meat of this chapter. We previously talked about the fact that one of the defining characteristics of neurons is that they are cells that can generate electrical signals. And now I'm going to talk a little bit more about what that exactly means and what it looks like. So I think, everyone has this kind of a vague sense of what electricity is. But if I asked you, what is actually traveling through those wires into your house to create electricity? The answer is charged particles. So when we talk about electrical signals being generated by neurons, we are talking about the movement of ions across the plasma membrane of those cells, which is going to change the charge inside the cell relative to the outside of the cell. Okay. So there's a term that we use a lot when we talk about neurons, and that term is membrane potential. So the membrane potential is the difference in the electrical charge between inside and outside the cell. So the difference in the balance of negative and positive ions. And the membrane potential of cells, particularly neurons, can change over time, right? Because ions can move in and out of cells. So we're going to be talking in this lecture about some stuff that hopefully you at least vaguely remember from back in Bio 151 when we talked about ion channels and how ions can use these channels to move in and out of cells. Okay, so the major players here, there are many ions inside cells and in the interstitial fluid. The major players here are going to be potassium and sodium, but other ions can also contribute to the generation of these electrical signals. But we're really going to focus mostly on those two because they are the most abundant ions inside and around cells. Okay, so all cells, including neurons, are going to have a lot of what are called potassium leak channels. So these are channels that potassium can pass through. And they're just openings. They're not, they have gates, but the gates randomly open and close. So we have kind of a steady leak of potassium across the plasma membrane. You also have a number of gated channels, which again, hopefully you remember this from Bio 151. These are channels that can be open or closed by specific stimuli. And the three types of gated channels that we've talked about are mechanically gated channels that open in response to physical force. So all the neurons that are involved in like your sense of touch, for example, so the reason why I can feel my hands touching each other are because I have in the neurons in my hands that sense pressure, I have mechanically gated ion channels that when I push on them are going to open up, and that's going to send a message that will go to my brain and make me feel something. You also have chemically gated ion channels, which are sometimes called ligand-gated ion channels. These two words mean the exact same thing. So this is going to open in response to some sort of chemical message or chemical in the area binding to the ion channel. So this could be neurotransmitters, hormones, or intracellular chemical messages, or they could also open or close in response to chemicals in the environment. So for example, if you eat something with chili peppers in it, right, there's a chemical called capsaicin that can activate neurons that are sensitive to it and cause ion channels to open, and that's what sends the sensation to your brain that causes you to feel that spiciness. Then there's voltage-gated ion channels. These are going to be very important for what we'll talk about the next lecture, but these are going to open in response to changes in the membrane potential, so the difference between inside and outside the cell. Okay, so now let's talk a little bit about what contributes to membrane potential when the neuron is, as we say, at rest. So the neuron is not in the process of generating an electrical signal. Okay, so I realize there's a lot of visual information here, but I just want to draw your attention to some key things. So this table is pretty important because it shows you the intracellular and extracellular concentrations of these important ions for maintaining the membrane potential. So I mentioned the big two are going to be potassium and sodium. So potassium is going to be more abundant inside cells in the intracellular fluid. There's a little bit of potassium in the extracellular fluid. For sodium, it's going to be the opposite. Most of the sodium is going to be in the extracellular fluid and very little in the intracellular fluid. This is something that you guys should memorize, okay? So most of this chapter, I would like you to sort of focus on understanding rather than memorization, but you will need to memorize the fact that sodium is more abundant outside the cell and potassium is more abundant inside the cell in order to be able to understand everything else that's going to happen with these electrical signals. Chloride is going to be less important. It too is also more abundant in the extracellular fluid than the intracellular fluid. What's really going to, though, be the major determinant, because the potassium and sodium kind of cancel each other out, the thing that is going to really contribute to the resting membrane potential is going to be the fact that inside the cell you have proteins, okay? In the extracellular fluid, in the interstitial fluid, you basically don't have any protein, and most of the proteins are going to be negatively charged. So the abundance of these negatively charged proteins inside the cell contribute to the resting membrane potential being about minus 70 millivolts, meaning that the inside of the cell has about minus 70, millivolts more negative charge than the outside of the cell. And by convention, we always refer to the outside of the cell as having a charge of 0. In fact, the outside of the cell can vary in its charge, but since what we're concerned with is the difference between inside and outside, we just call the outside 0, and then we talk about the inside relative to the outside. Okay, so at rest, the inside is going to be about minus 70 millivolts because of those negative proteins. And then you can see here, this figure shows just all of the different membrane proteins that are going to contribute to maintenance of the homeostasis of this. So we have, of course, the sodium-potassium pump, which you guys should remember well from Bio 151, which is going to contribute to there being high sodium and high outside the cell and high potassium inside the cell. Then there's these potassium leak channels. We can see there's a lot of them. Here's also a sodium channel, but there's not as many of those. So the cell is much more leaky to potassium than it is to sodium. Okay, so then when we think about ion flow and what's going to happen when we open sodium channels or open potassium channels, so potassium is more abundant inside the cell. So if you open a potassium channel, if you have a gated channel that opens potassium channels, potassium will flow out. Sodium is more abundant in the extracellular fluid. So if you open up a sodium channel, sodium is going to flow into the cell, okay? So because you are, when you open sodium and potassium channels, moving charged particles either into the cell or out of the cell, that's going to change the membrane potential when you do that, okay? So when you change the membrane potential, you either what's called depolarize the cell or hyperpolarize the cell. So you've probably heard the word polarize or polarizing before, or even the word polar, right? So polar means like opposite. So depolarizing means making two things less different, right? Polarizing means it's like in something that people have really strong differences about, right? So depolarizing means making the differences smaller. Hyperpolarizing means making the differences bigger, okay? So what these graphs show is changes in the membrane potential in response to ion movement. So as I mentioned, right, this is a typical cell and it's going to start out at about minus 70 millivolts. So this is the cell under resting condition, okay? And so this is going to show what's going to happen if you have stimuli that are going to open potassium channels, okay? So you'll recall potassium is going to be more abundant outside the cell, inside the cell than outside the cell. So if you open potassium channels, potassium will leave the cell. Potassium is positively charged. So when it leaves the cell, it's going to make the inside of the cell more negative, right, relative to the outside. If you're taking positive ions and you're removing them from the inside of the cell, which was already more negative than the outside of the cell, you are going to hyperpolarize the cell. You can see here that the membrane potential becomes even more negative than it was initially. And then eventually, with the help of the sodium-potassium pump, you'll go back to normal, okay? The opposite is going to happen if you create conditions where you're opening sodium channels. So remember, sodium is more abundant outside the cell. So when you open sodium channels, sodium is going to go into the cell. Sodium is also positively charged. So when sodium comes in, it's going to make the cell less negative, closer to having no difference in charge between inside and outside the cell. So we can see that happening here. We can see that when sodium channels are opened, sodium, positively charged, will go into the cell. That will make the difference between inside and outside the cell less, or depolarizing the cell. Okay, so hyperpolarization means making the inside and outside more different in terms of charge. Depolarization means making the inside and outside less different. Okay, so again, potassium, when you open potassium channels, potassium will tend to leave the cell, and that will hyperpolarize the cell. Sodium, when you open sodium channels, sodium will enter the cell. That will depolarize the cell. Okay, so when either of these events happen, that can result in the generation of two different types of electrical signals. So one type is called graded potentials. We're going to talk about those right now. The other type is called action potentials. We're going to talk about those in the next lecture, but that's what this third picture is showing here, what happens when an action potential gets triggered. But right now we're going to just stick with graded potentials. Okay, so again, potassium is more abundant in the intracellular fluid. When potassium channels open, potassium is going to flow out. That will hyperpolarize the cell. For sodium, it's more abundant in the extracellular fluids. When you open sodium channels, sodium is going to flow in, depolarizing the cell. I realize I've repeated myself like several times here. That's because this is important. And if you don't have this down, it's going to be really hard to figure everything out. So you really need to kind of commit this to memory and have this solid, or else nothing else is going to make any sense. Okay, so graded potentials, right? Graded potentials are going to be changes in membrane potential where you have opening or closing of ion channels, and it's going to generate an electrical signal in the cell because ions are flowing in or out. The size of the graded potential, the amount of difference in the membrane potential is going to be proportional to the strength of the triggering event. So it could be like 50 ion channels are open or only one, right? So if only one's open, you're going to have a smaller change in the membrane potential. Another thing about graded potentials is that they are going to initiate at one specific spot on the neuron. And so if you're looking very close on the membrane of the cell to where that ion channel opened, you might see a very big difference. But as you get further and further away, the effects of the graded potential are going to decrease as you travel further and further away from the site where the ion channels opened and closed. So they're going to sort of wear out as they travel across the cell. And then graded potentials also, because they can involve any type of ion, right? They could involve potassium, they could involve sodium, they could involve chloride, they could involve any of the other ions in the cell. Depending on what's happening, they can either depolarize the cell, right? So they can make it less negative, or hyperpolarize the cell. These are both examples of graded potentials. Okay. Another thing about graded potentials that's important to understand is that if you have two graded potentials happening close to the same time, you can actually have their effects on membrane potential end up being what's called summed or added together. What's shown in this graph here is two different graded potentials happening in the cell. So there was one stimulus that caused some ion channels to open, and that caused a depolarization event. Then there was a second stimulus that caused some other ion channels to open, which also caused a depolarization. So the dotted line represents what would have happened if these stimuli had happened just in different cells, independent of each other. But you can see that the ions that had entered the cell from stimulus 1, a lot of them were still there when stimulus 2 occurred. So you can see here, this is the summation. So the difference between the purple up here and the dotted line down here represents the effects of adding together these two graded potentials. So if you have two signals happening at close to the same time and close to the same location, they can be added together and they can have a greater impact than either one of them individually.